Picture this. You have a dog. Or you've had a dog, or you know someone who has a dog, or you've at least encountered the concept of dogs enough to know how the timeline goes. You get a puppy. It is small and chaotic and enthusiastic about existence in a way that makes you feel slightly ashamed of your own energy levels. You raise it. It becomes an adult dog in about a year. It is your companion for a decade, maybe a little more, maybe a little less, depending on the breed and the luck of biology. And then it is old. And then it is gone. And you are still here. Substantially less chaotic than the puppy was, possibly even more chaotic in certain ways, but biologically you have barely gotten started. You will live, if the actuarial tables are to be believed, approximately 6 to 7 times longer than that dog. The dog is not a bad animal. It is not living badly. It is not being punished by evolution. A dog's lifespan is entirely appropriate for a dog. It fits within the expected range for a mammal of its body size and metabolic rate, following rules that apply consistently across most of the mammalian world. The dog is, in a statistical sense, normal. You are the weird one. The human lifespan is one of the most anomalous data points in mammalian biology. By every standard prediction model that uses body size, metabolic rate, and phylogenetic position to estimate expected lifespan, humans should live somewhere between 20 and 40 years. We are instead living to 70, 80, 90, and beyond with what, from an evolutionary perspective, is almost suspicious regularity. Something happened in the human lineage that broke the standard mammalian aging rules, and figuring out what that something was and why it happened is one of the most fascinating questions in the biology of aging. Start with the rules that humans are breaking, because you can't appreciate the exception without understanding the pattern. Across the mammalian class, there is a remarkably consistent relationship between body size and lifespan. Small mammals live briefly. Mice live 2 to 3 years, rats live 3 to 4 years, rabbits live 8 to 12 years, large mammals live longer, elephants live 60 to 70 years in the wild. Whales live even longer. Bowhead whales have been aged at over 200 years, making them the longest-lived mammals known. The relationship is not perfectly linear, but the correlation is strong and consistent. Bigger animals generally live longer. The underlying mechanism for this correlation runs through metabolic rate. Small animals have higher metabolic rates per unit of body mass than large animals. A mouse's heart beats approximately 500 times per minute, compared to a human's resting rate of 60 to 80. High metabolic rate means faster oxygen consumption, which means more oxidative stress, the cellular damage caused by reactive oxygen species produced as byproducts of aerobic metabolism. Higher oxidative stress accelerates cellular damage, which accelerates the biological processes associated with aging. A mouse lives fast in the metabolic sense, and accordingly, dies young. Applying the body size and metabolic rate formula to humans produces a predicted lifespan of approximately 20 to 40 years, consistent with what we observe in other primate species of similar size. Chimpanzees, our closest relatives, live approximately 40 to 50 years at maximum in the wild, with many dying younger from disease, predation, and injury. Gorillas live to approximately 35 to 40 years in the wild. Orangutans, somewhat longer, reaching perhaps 45 under good conditions. These are the numbers the standard mammalian formula predicts for a primate in the human size range. Modern humans routinely live twice as long. Traditional hunter-gatherer populations, often cited as the closest living analog to pre-industrial human lifespan, show maximum lifespans in the 70 to 80-year range when individuals survive the high childhood mortality that characterizes pre-modern environments. This means that the longevity advantage is not primarily a product of modern medicine or sanitation. It is a biological feature of the human organism that predates modern civilization. The grandmother hypothesis is the most influential evolutionary explanation for why humans live so far beyond their reproductive years, and it is one of those ideas that seems almost embarrassingly simple once stated, but that has profound implications for understanding human biology. In most mammals, the end of reproductive capacity and the end of lifespan are closely correlated. Female mammals typically die within a few years of losing fertility, the evolutionary logic being that if an individual can no longer contribute genes to the next generation through direct reproduction, selection has little reason to maintain that individual's survival. Resources spent maintaining an aging post-reproductive body are resources not spent on reproduction or on supporting currently reproducing individuals. Human females undergo menopause, complete cessation of reproductive function, typically in their late 40s or early 50s. They then regularly live another 3, 4, or 5 decades. This is unusual among mammals. Killer whales and short-finned pilot whales are the only other species known to undergo genuine menopause and survive substantially past the end of fertility. The question is why? The grandmother hypothesis answers this by proposing that post-reproductive women in traditional human societies made substantial contributions to the reproductive success of their daughters and the survival of their grandchildren through food provisioning, childcare, knowledge transmission, and social support. A grandmother who helped her daughter raise children to reproductive age was contributing to the survival of her own genes even without directly reproducing herself. If grandmotherly assistance substantially increased grandchild survival rates, and field studies of traditional societies suggest it did, then the genes contributing to longevity past menopause would be favored by selection. This creates a feedback loop. Longer-lived grandmothers helped more grandchildren survive. The grandchildren carried the grandmother's longevity-associated genes. Those genes spread in the population, and human lifespan extended progressively beyond what the standard mammalian formula predicted. The evolutionary driver was not any single genetic change, but the cumulative selection pressure of a social and subsistence system in which post-reproductive knowledge and labor added measurable reproductive value to the lineage. The hypothesis has attracted both support and criticism in the decades since its initial formulation. Supporting evidence includes the documented beneficial effects of grandmaternal presence on grandchild survival in multiple traditional societies, the consistent pattern of menopause appearing at roughly the same absolute age across diverse human populations, and theoretical models showing that grandmothering behavior can mathematically account for the degree of human lifespan extension observed. The extended juvenile period is both a consequence and a cause of the human longevity pattern, and understanding it clarifies why the whole system hangs together as an evolutionary package. Humans have the longest period of childhood dependency of any mammal. A chimpanzee is weaned at around 5 years of age and reaches sexual maturity at around 10 to 13 years. A human is typically weaned at 2 to 4 years, but doesn't reach sexual maturity until 12 to 15, and isn't considered socially and cognitively adult by any human cultural standard until considerably later. The time between weaning and first reproduction in humans is strikingly long. This extended juvenile period has costs. A long dependent childhood means a long period of intensive parental and social investment per offspring, high metabolic cost of supporting non-reproducing individuals, and delayed return on the biological investment in producing offspring. The costs must be offset by corresponding benefits, and those benefits are cognitive and social. The human brain grows and develops across a much longer time window than any other primate brain. Brain development in humans continues into the mid-20s. Prefrontal cortex myelination, the structural completion of the neural circuits governing complex decision-making, continues long after most other mammals have fully mature nervous systems. This extended development window allows for the acquisition of an enormous amount of learned knowledge and skill. The cultural inheritance that each human generation passes to the next, and that is too complex to be encoded genetically or learned in a short developmental period. The extended juvenile period and the extended lifespan are thus related adaptations. Long life provides the time to accumulate the extensive learned knowledge that long life in a complex social environment requires. Extended childhood provides the developmental window for the brain to grow large enough and complex enough to store and process that knowledge. Each adaptation makes the other more valuable, creating a package of traits that reinforces itself. The cellular and molecular mechanisms by which humans achieve their extended lifespan relative to similarly sized primates are now reasonably well characterized, and the picture they present is one of a species that has evolved enhanced maintenance systems across multiple dimensions simultaneously. DNA repair capacity is one of the most important factors in organismal lifespan across species, and humans have substantially enhanced DNA repair mechanisms compared to shorter-lived primates. Across mammalian species, DNA repair capacity correlates with lifespan more strongly than body size, or metabolic rate does. Longer-lived species have systematically better maintained genomes. The accumulation of DNA damage in somatic cells is one of the primary drivers of cellular aging and the loss of tissue function that characterizes aging organisms, and species that invest heavily in repairing that damage stay functionally younger for longer. By the way, if you want to go even deeper into the secret lives of ancient humans, I just launched a full course called The Secret Lives of Ancient Humans. 10 modules, 3+ hours of content covering everything we never talk about on YouTube. What they feared, how they communicated, what hunted them, and much more. Plus an exclusive PDF guide included. Link in the description. See you inside. Telomere maintenance is a related factor. Telomeres are the protective cap sequences at the ends of chromosomes that shorten with each cell division, eventually reaching a critical length that triggers cellular senescence or death. Telomere length and the activity of telomerase, the enzyme that rebuilds telomere length, influence how many times cells can divide before reaching this limit, and this limit contributes to the aging of tissues that require ongoing cell replacement. Human telomere biology is calibrated for longer potential cell division cycles than that of shorter-lived primates. Antioxidant systems are the molecular defense against the oxidative damage that metabolic processes continuously produce, and humans have several antioxidant mechanisms that appear more robust than those of shorter-lived mammals. Superoxide dismutase activity, glutathione metabolism, and other components of cellular antioxidant defense show patterns across mammalian species that correlate with lifespan, and humans fall at the high end of these correlations relative to their metabolic rate. Cancer suppression mechanisms are particularly relevant because cancer, the dysregulation of cell division, is fundamentally an aging phenomenon, with risk increasing dramatically with age, as accumulated somatic mutations and cellular quality control failures allow malignant clones to emerge and proliferate. Larger animals with more cells should, on statistical grounds, have higher cancer rates than smaller animals. More cells means more potential cancer initiation events. But this expected correlation doesn't hold empirically. Elephants have extremely low cancer rates despite having 100 times more cells than humans. Whales, with even more cells, have similarly low rates. The mechanism in elephants involves multiple copies of the TP53 tumor suppressor gene. While humans have 2 copies, elephants have approximately 40. Similar enhanced cancer suppression mechanisms presumably exist in other long-lived large mammals. The immune system's role in longevity is a dimension of the biological picture that has received increasing attention, and that helps explain why extended lifespan required coordinated evolution across multiple physiological systems rather than a single change. Aging is partly an inflammatory process. Chronic low-grade inflammation, sometimes called inflammaging in the gerontological literature, is a consistent feature of the aging mammalian body, and it contributes to the deterioration of multiple organ systems over time. The immune system, as it ages, loses precision and gains dysregulation. It becomes less effective at targeting actual pathogens and more prone to generating inflammatory responses to self-derived signals that it misidentifies as threats. Human immune systems have several features that appear calibrated for extended functional maintenance compared to shorter-lived primates. The distribution of immune cell types, the balance between pro-inflammatory and anti-inflammatory signaling, and the molecular mechanisms that regulate immune senescence all show patterns consistent with selection for extended immune function. This is not a guarantee of blastart. The immune system does decline with age in humans, just more slowly than the baseline mammalian pattern would predict. The interaction between the immune system and the microbiome is another relevant dimension. The gut microbiome, the community of bacteria, archaea, fungi, and other microorganisms inhabiting the human digestive tract, influences immune function, inflammatory tone, and metabolic efficiency in ways that are increasingly understood to affect longevity. Human gut microbiome composition changes with age, and some of those age-related changes correlate with markers of accelerated aging and increased disease risk. Centenarians, individuals who live past 100 years, show distinctive microbiome compositions that differ from those of shorter-lived elderly individuals, suggesting that favorable microbiome maintenance may be one of the biological factors contributing to exceptional longevity. The brain's role in human longevity is underappreciated, but may be one of the most important dimensions of the entire story. The brain is the organ that most directly enables the behavioral and social patterns that make extended human lifespan evolutionarily valuable. Without the cognitive capacity to accumulate, store, and transmit the cultural knowledge that makes grandmotherly assistance and intergenerational knowledge transfer valuable, extended post-reproductive survival would offer little evolutionary payoff. The brain's capacity for complex learning and social cognition is what makes old human beings worth keeping alive, from an evolutionary standpoint. But the brain also directly influences longevity through its regulation of systemic physiology. The hypothalamic pituitary axis, the stress response systems, the neural regulation of immune function, and the brain's role in metabolic regulation all mean that the brain's functional state has downstream effects on every other organ system's aging trajectory. A brain that ages well tends to support better maintenance of peripheral organ systems. A brain experiencing chronic stress, through sustained activation of the cortisol stress response, accelerates aging in peripheral tissues through several mechanisms including immune suppression, glucose dysregulation, and direct cellular damage from cortisol excess. The social buffering of stress that human social groups provide, the documented reduction in cortisol responses to stressors in the presence of trusted social partners, is therefore a genuine biological mechanism through which social connection influences longevity. The observation that socially isolated individuals have shorter lifespans than socially connected individuals, which is one of the most consistently replicated findings in the epidemiology of aging, has at least part of its biological mechanism in the stress hormone buffering that social connection provides. The evolutionary timeline of human longevity extension is difficult to pin down precisely because lifespan leaves no direct fossil trace, but the indirect evidence provides some guidance. The changes in human anatomy that correlate with the grandmother hypothesis and the extended juvenile period framework, the reorganized female pelvis reflecting the obstetric challenges of large-brained infant delivery, the reduced canines and face reflecting dietary changes, the leg anatomy reflecting committed terrestrial bipedalism, these changes occur primarily in the transition to Homo erectus and the subsequent elaboration of the Homo lineage across the past 2 million years. Paleodemographic analyses of hominin populations from skeletal age indicators, examining the distribution of ages at death in fossil assemblages to infer something about population age structure, are methodologically challenging, but have suggested that the proportion of individuals surviving to older ages increased over the course of hominin evolution. Homo sapiens skeletal assemblages show higher proportions of older individuals than earlier Homo species, consistent with the progressive extension of lifespan across hominin evolution, rather than a sudden change at the emergence of anatomically modern humans. The genetic evidence for longevity-associated variants in modern humans provides another line of evidence about timing. Several genetic variants associated with longevity in modern human populations, variants in genes related to DNA repair, inflammation regulation, lipid metabolism, and other aging-relevant pathways, show signatures of positive selection that date their selective sweep to periods consistent with the general framework of human evolution over the past 1 to 2 million years. The comparison with other exceptional mammalian longevity cases provides perspective on how remarkable the human case is and what it shares with other long-lived outliers. Naked mole rats are the most frequently cited rodent counterexample to the standard body size lifespan relationship. They weigh approximately 30 to 35 grams, comparable to a large mouse, but live up to 30 years, approximately 10 times longer than the standard mammalian formula predicts for an animal of their size. Their longevity is associated with unusually robust DNA repair, exceptional cellular quality control through enhanced proteasome activity, a distinctive metabolic profile involving resistance to oxidative damage, and potentially related to their social structure. They are eusocial, with a reproductive queen and large numbers of non-reproducing workers, resembling in some ways the social structure that the grandmother hypothesis invokes for humans. Bats are another systematic outlier. Many bat species live far longer than the body size rule predicts, with some species reaching ages of 30 to 40 years despite weighing only a few grams. Bat longevity has been associated with enhanced antioxidant systems, unusual DNA repair capabilities, and distinctive immune system properties that allow bats to harbor viruses without the inflammatory damage those viruses cause in other mammals. The common thread across exceptional mammalian longevity cases, humans, naked mole rats, bats, whales, is the combination of enhanced molecular maintenance systems, DNA repair, antioxidant defense, protein quality control, with particular social or ecological circumstances that made extended longevity evolutionarily valuable. Longevity is not free. It requires investment in maintenance systems that is only worthwhile if the extended lifespan actually produces evolutionary payoffs. Where those payoffs exist, evolution apparently finds the molecular solutions to make longer life biologically possible. The practical implication of the human longevity anomaly for modern life is worth addressing directly, because understanding why humans live longer than standard mammalian prediction has concrete implications for how that longevity is best maintained. The maintenance systems that evolution equipped humans with for extended lifespan, enhanced DNA, repair, immune regulation, antioxidant defense, social stress buffering, were calibrated for a particular lifestyle: sustained physical activity, diverse whole food nutrition, social embeddedness in groups providing genuine mutual support, meaningful cognitive engagement across the full lifespan, adequate and regular sleep. These were not the conditions of a privileged minority in ancient populations. They were the conditions of ordinary human subsistence life. Modern environments diverge from these conditions in ways that are often significant. Chronic sedentariness, processed food consumption, social isolation, chronic stress from non-physical stressors that activate the cortisol system without physical resolution, and sleep disruption from artificial light and schedule demands all represent mismatches between the conditions for which human longevity maintenance systems were calibrated and the conditions in which those systems are now operating. The result is that the biological potential for human longevity, the maintenance systems evolution built, is not being equally expressed across all modern populations and individuals. The remarkable longevity that the human lineage evolved to achieve is not a guarantee written into the genome, but a capacity that depends on the conditions of its expression. There is something both humbling and clarifying about understanding human longevity in evolutionary terms. Humbling because the extra decades of life that humans carry above the standard mammalian formula are not a gift from nowhere or an entitlement of the species. They are the product of millions of years of selection operating on the specific social and cognitive circumstances of the human lineage. The value of accumulated knowledge in a species that learns rather than instincts rather than instinct. Executes the value of post-reproductive assistance in a species whose offspring are so demanding that raising them requires more than 2 adults working alone. The value of extended cooperation in a species whose survival strategy centers on collective action. Clarifying because it tells you what the long life is actually for. It is not for retirement or for its own sake. It is for transmission, for the passing of accumulated knowledge, skill, and social capital from those who have it to those who are building it. The grandmother carrying children while the mother forages, the elder who knows where water can be found in the 3rd year of drought, the experienced hunter who can read animal behavior that younger hunters can't yet see. These are the evolutionary reasons that old humans were worth keeping alive. That function hasn't changed. The specific content of what experienced older humans know has changed dramatically. But the underlying logic that a species whose survival depends on accumulated cultural knowledge benefits enormously from individuals who have accumulated a lot of it and can share it, remains as valid in the current era as it was in the Pleistocene. The dog gets 7 years, give or take. You get 70 or more. That discrepancy is not accidental, not arbitrary, and not simply a matter of body size. It is the biological record of a species that evolved a particular solution to the problem of surviving in a cognitively demanding social world, and then lived long enough to write about it and wonder why, which is, taken altogether, a very human thing to do.